All right, guys, real quick, before we get started, what we're going to be looking at today is what does DNA look like? So that's our essential question for today. I put a real quick bell ringer together for you guys. So what I'd like for you to do is use your smartphones, your tablets, whatever you have with you today. Share if you need to. Answer these two quick questions for us, and it's going to build on what we're looking at today with DNA. Okay? Any questions?
No.
All right, let's go.
Isn't it always a double helix? Or is that what it looks like all together? You mean individually?
Just what the structure looks like. What do you think the structure is going to look like? When you see it, what's it going to look like?
A clump.
Okay.
Can I resubmit my answer?
Yes.
How do you do that?
Does it
Go.
say submit another one?
No.
Yeah.
Do
Can I
that.
submit another one by my name?
Yes.
Okay.
Yes.
Like spaghetti.
Like spaghetti.
Where is DNA found? In everything.
Hair follicles and everything in blood. And
Except like tables, right? Because that's cells. There's something that's in everything. Isn't that cells?
atoms.
Atoms. Thank you, sir. Okay. Hair. Oh, okay. You know a lot about that one.
That's the question you're looking at? Where?
I wish I could.
That would be. Would need to be the next. That's the question you're looking at? Where?
I wish I could.
That would be. Would need to be the next. Technically incorrect, but accurate.
I don't have any.
But that's just accident. Chromosomes and DNA.
In our cells.
You were
Forgot
close.
about cells.
Where you go?
Sting my eyes.
And I look at my partner where I got an 85.
Practice screen.
I can see the name is Michael.
During the ball ring.
I never tried so hard to make
How do you carve
That's too
Every element technically.
Okay,
We
so
were about to turn.
now that we looked at that and answered those questions, let's talk for a couple minutes. Bevan, you with me? Thank you. Okay, so the standard that we're going to be looking at today is students will analyze the nature of the relationships between structures and the functions in living cells. Okay? If you remember, we've been talking about all year how structure follows function, that everything is designed the way it is because of the job it has to do. So when we look at something's structure, it tells us a lot about what its job is. So the structure we're going to be looking at today obviously is DNA, and we're going to be analyzing it so that we can see the relationship it has to the job that it actually does in the body. Okay? So we're going to do a little hands-on thing today where we're going to actually extract the DNA from some strawberries and look at it. Anybody have an idea about why I picked strawberries?
Because you could, it was something about because you could see it or something like that. Because I know because we did it.
Carolyn?
Never mind.
Okay.
Seeds?
It got
Um,
something to do with the little holes on the outside, right?
The seeds?
No. Okay. Holes.
Strawberries are what we call octoploid. In other words, they've got 8 times as much DNA as normal things do. So it's a lot easier for us to see because we're going to be able to generate a lot of it. Okay? And we're going to use 2 things. The first thing we're going to use is we're going to use what's called a DNA extraction buffer. This is basically just water with soap and salt added to it. And the salt has a job and the soap has a job. Basically, the job of the soap is to break down that phospholipid bilayer that surrounds the cell and surrounds the nucleus that holds the DNA inside. And then the salt's job is to kind of break down the proteins that are around the nucleic acids. So we'll actually be able to see it. Well, we'll be able to pull it out of the cells of the strawberry. And then we're going to use ethanol. And the reason we use ethanol is because DNA is not soluble in ethanol. Right? So in other words, it won't dissolve in the ethanol. It'll actually, you'll see it's going to be kind of cool. I'm not going to tell you what's going to happen exactly yet, but when you pour the ethanol in, there's going to be something really cool that happens. Okay? Any questions?
No.
Okay, I have one set up here for each table. You guys are already in the groups that I put you in. Remember, everybody needs a pair of goggles. They're in the back cabinet where we always keep them. So send one person from your table back to get goggles and send someone else up here to get equipment. Okay? Yes, ma'am. Yes, I'm getting ready to pass them out right now.
That's the cool guy. Somebody got the other guy.
Guys, remember, clean your desk off for me from all your stuff except for what you're using.
Berries.
50 million test tube, a glass rod.
Strawberries. The test tube. You gotta grind it up.
Test tube. This is a glass rod, right? 10 milliliters of
This is the rod. This is the rod. This is the rod. You start with this. This is test tube. That's the graduated system. Let's go put a strawberry in.
This is
2 minutes.
2 minutes?
Yes.
2 minutes.
Ethanol is in the ice.
We want to make sure that we follow the teacher's instructions.
Yeah, let me see your phone so I can type it.
I want to use a pipe.
Yes.
I probably got like a minute left now.
I bet she probably got a cold on it too.
Don't touch my phone. We have a leak. Well, maybe you should be smashing up that high. We wouldn't have a leak, boy.
How you lick?
Should lick it. There's a leak. That's why Dawn should have did it.
No. That would have been like a baby who didn't like it smashed peas and had
Careful not to break the bag, Lauren.
When?
Probably Saturday or Sunday.
Alright, so did you finish mushing it? Probably bring it all to the bottom now so we can put the stuff in there.
We did keep a track of how much we're pushing
And then I had to stay after school and
Go ahead, don't get that on me.
and now let's follow
Oh,
the
this is
label
just salt.
instructions.
Yeah, don't get that on me. Let's go.
You can drink it and be just fine.
You're funny.
Careful.
Really funny.
That's all?
No, this is you. No, this is Lauren's
Come
job.
on, pour it, pour it.
Yeah, I should need your phone now.
That's good.
Wait, don't start mushing it.
Come on,
You
y'all.
have to mush it for a minute.
Is it closed?
Yes, it's closed.
Just start. Just go ahead and start. Knead or mush?
Pour it in. Yeah, just pour it on top. Like right here.
Okay.
Can't get it open. There you go.
You're gonna put it in this.
And then wait.
Put it in the. You're gonna put it in the thing.
Go ahead and go get the ethanol. Before I pour? Yeah, because it needs to be in here. The ethanol needs to be in here. Yeah. How much ethanol do I need? 50? 20.
So we need
20? 20 milliliters.
So we need to put ethanol in here first.
Good thing we caught that one.
We put the strawberry in there first.
Oh, first?
Well, no, you're gonna strain it first. Huh?
Yeah, into here and then
So I was right. All right.
I really should have got myself.
That looks yum yummy.
Lower the cloth.
That was a fast career.
You are not, who is that? Main Divine. Please, you're not retired.
They good?
Yeah. I think you're right.
Yeah, you want the phone.
7 seconds, so it's not that bad.
Ketchup. Alright. Now we need some ethanol. Is that the ethanol?
Mm-hmm.
Alright. Let it drip into the juice.
Back.
I think you gotta put this together first.
Yeah.
Let it filter.
I'll give it more area to drip through. Strawberry mush.
Alright, pour it in.
No.
You want all of it in there?
Yes, squeeze it all.
Can I just like
Speed up the process here. We need to
Fruit strawberry.
No, you don't need to.
Yeah, you ready?
Where's your cheesecloth?
Yeah.
Where'd it go?
That was Trey's responsibility.
It's on the table up there, so run get one of yours.
Squeeze the rest of it.
I don't want to squeeze it.
It doesn't need to be squeezed. You're okay.
want to support through the mesh cloth?
The ethanol now needs to be poured through the mesh as well. Yes.
So with the strawberry.
Yes, it gets poured right over the top of that. Okay, so but make sure you open it back up.
Okay. Okay.
Either pick it up either.
Good work, you guys.
Pour the strawberry slurry in. No, you need to do that. Okay.
Typically serve this.
Some of your filtration on. They said pour the strawberry slurry. Yeah. Pour the strawberry.
Just put it down.
Slurry.
The milk of glass rod into the two-way strawberry extract and ethanol layers come into contact with each other. You can move it over the bag, actually.
What was that on phone?
No, it's supposed to be where they come in contact, not the bottom.
I'm so serious.
Right there, where the strawberry and the ethanol are. Wow. That's cool.
That looks gross.
That's pretty neat.
That's pretty cool. That's the DNA.
That's the DNA. Record those observations.
Remember? No, it's the name of Frank Oklahoma. And you know that it's the name of my PowerPoint about freaking.
When you go to Malik,
Stop it.
when you get to the part where you're going to put the inoculating loop down in the test tube, just very slowly stick it in there and don't stir yet, because what I want you to see is when the precipitation happens, what it looks like before it gets all twisted around. All right, so when you first do it, just kind of stick it in there and see what happens. You know, make a good observation of what's going on when you first put the inoculating loop in there. And then once you do that and you see it, then you can wrap it around, pull it out of there if you want to, so you can look at it better. But I want you to see the shape it looks like when it initially precipitates, okay?
Use like a small double helix.
Yes.
If it does not look like a small double helix, I'm gonna be upset.
Have you got the ethanol yet? No. Go get the ethanol. You're going to pour the ethanol right on top of it.
Oh, that makes it good fast.
But you see the difference between the water and the ethanol? It's cool, man.
You ready?
What's the thing with the milk and the
Pull it out.
toothpick
Yeah.
and the food coloring?
Wait, did we already pour the stuff in
Yeah,
there?
the ethanol's already in there.
No. osmosis and you up where you
Pretty cool.
Have you got it? Yeah. So look, can you see it? I mean, I want you to really see what it looks like. Can you see how
It looks like it's turned into like foam or some type of.
That's cool. So this is it?
Yeah, so you see, if you look, you can see the very ends where it's like almost individual strands. See how it's kind of curled out like this? Mm-hmm. You can almost kind of see the form where how it's shaped into that spiral double helix, right?
Okay, so I'm not
Yeah,
convinced.
That's a lot of DNA.
You would have to have a microscope to see that.
Yeah, you really would have to have a microscope. It's very small.
There's more down here.
The strawberry is the only type of thing that can do this.
No, you can get DNA out of anything like this.
No, but I mean
except-
like that much.
No, there are other plants that are. We could have used banana also and got almost as good a result with banana as well. But there are other plants that are octoploid like that where you get that. But strawberries is just kind of the easiest to do.
So Mr. was like,
There's a lot.
all right.
Now, which one for? Which one?
Yeah.
Okay, guys, listen up. Guys, listen up. If you are finished, if you've already extracted the DNA, go ahead and start working on the answers on the back of the sheet, and then as soon as everybody's done, we're going to discuss what we just saw, okay?
Yes, sir.
You see that little buddy?
No, but yeah, but I don't understand though, Lauren.
I'm waiting for Lauren to think of it. I was like, what am I waiting on?
You gotta observe first, okay? Are you done observing?
More, and I don't think there's anything more to observe.
Are you sure?
Yes.
Dip the loop or glass rod into the tube where the strawberry extract.
Don't break like that.
Here, I'm not doing it.
Yeah, just put it in there. Don't stir it around yet.
Well,
What I did just now,
we can do this one day when we get.
I feel like
Okay, am I done here? Wrong side, wrong side, wrong side.
No, there's nothing else to do. Okay, then go ahead and swirl it around.
What's up?
Lift it up.
You want to see ours? Ours was greater.
So that's the
That's the DNA.
Hey, it looks like a tornado's coming off the tube.
Oh my god. Come here.
Ooh, tornado hunters.
that's cool.
Hey.
It's like perfect.
It's like one of them little gel things.
You didn't get the glass thing and they put the pictures in it?
They put that.
Yeah, but if you see just like the very ends, these little bitty strands, you can kind of see them just curling up just a little bit. So you kind of get an idea of what that structure looks like, even though it's microscopic. You can kind of start seeing it. But that, y'all got a lot. That's really good.
Awesome sauce.
Awesome sauce?
Awesome sauce.
Okay, I'm done.
Talk about putting it in
Oh no, I was like.
Okay, so everybody's
That's a lot of DNA for one little strawberry.
Now eat it. No.
Okay, that's done.
What'd you get? Lauren, I didn't finish. I'll make sure you read it.
Oh, excuse me.
Actually, off the DNA and the back thing.
Aw, man.
You did it backwards?
No,
Yes.
I just.
Yeah, because
I got confused. Shut up.
you did it backwards. Is there DNA in your food? Yes. How do you know? Because strawberry is food and it has DNA in it.
have to get kind of to really be able to see it. I don't have a microscope that's really strong enough to be able to see that. So, Amaya, what did it look like? What did you see?
The DNA was curly at the end, and it kind of looked like egg whites
Okay, so
or mucus.
if you think about it, from what we know about structure of DNA from studying in class, does it make sense that it was kind of curly?
Yes. Because the double helix spins.
Right, the double helix is in a twist, so absolutely. So the question came up when we were standing up here is, what would happen if we stuck it in the microscope and looked at it? Would we be able to see that? The answer is no. Okay, first of all, I don't have a microscope that is strong enough for us to actually see it because it is so small. And also, the second you stuck that inoculating loop in there, and it kind of precipitated, it immediately kind of clumped all together. And so it would be very hard for us to kind of separate those strands out into an individual strand and get a good look at it. But isn't that really cool? I mean, you guys saw DNA, right?
Yeah.
DNA is what makes us who we are, what makes everything what it is. And we were just looking at it and holding it in our hands, basically. Okay? So what I want you to do is I want you to finish up the lab report part of this, and then for an exit ticket today, what I want you to do is go back to those original 2 questions from the bell ringer. What does DNA look like? And I just want you to write a short summary on what you saw and what you think DNA looks like. Okay?
Okay.
All right, thanks, guys.
Okay. Make sure her multiple choice is right.
My multiple choice is D, A, C, B.
No, it's DCB.
No, it's Lauren. We went back in the beginning paragraph and it said.
Let me just send a text message and I'll put my name.
Yes, sir.
Do we have to do it?
Yeah, I should have it from there.
Remember when I first talked about what was going on while we were using the buffer, the extraction buffer?
It's to separate, it's to break open the cell.
So what was the job of the soap?
To break it open.
Right, to break down the biophosphate
For the cells?
layer. For the cells and the nucleus, right? Both. So it's breaking open the cell, right?
Yes.
That's what okay, so
Okay, so now it just
this
looks
is
wrong.
D.
Okay, so the B is the one with the
And
soap,
this is
which is
A.
breaking open the thing, and then D.
Wait, no, that's not
This
A.
is A.
So is
This is
it
C.
Okay.
DCBA.
What does
that was the
that?
filter the strawberry through the cheese?
No,
That was the
ethanol.
ethanol.
That was the grinding.
Wait, which
the
were you
smashing.
at?
Let's look at the bottom one first.
Oh,
break
break
up
up.
proteins and dissolve cell membranes.
The initial smashing and grinding of
Oh,
strawberries.
D. The soapy sausage is
Right, the soapy solution. Yeah. Because remember the salt dissolves the proteins, and the soap.
break down the.
Right. Yeah, you got it. All right.
So it's C, D.
No, D has to be
So
to
A
precipitate.
is the second
This one
one.
had to be A.
The second one is A.
So that means this one had to be D.
No, the first one still has to be D, right?
No.
It's C, it's C, A, D, B.
D is the third one. It's
That
This
doesn't make
is the very
sense
last
because the
thing
D,
done,
yeah,
right?
So that means what makes it the same?
Right, like an analogy.
Correct. Yeah.
Okay, when you wild cotton into a rope, obviously it's clumped together, but the only way DNA gets clumped together is when it's in ethanol because it's not soluble.
I mean,
How does that
Well,
relate?
it can be clumped together in different ways too. But
It also think about what's going on when right before cell division happens, when the DNA is getting ready to split in the nucleus. Think about what happens with DNA production in the nucleus, and just think about some of those things, right? Because there are other times that the membrane around the nucleus breaks apart, right, and the DNA comes out.
Okay.
Right?
Okay, so number 5 is why is it important for scientists to be able to remove DNA?
Yes.
So for one, they're going to study and extract data from it, but like what kind of data would they extract? Like, would you see like hereditary things?
yeah, wouldn't it be like
Absolutely, that would be one thing.
Like hereditary traits that they
Yes.
inherit?
Yes, most definitely. That would definitely be one thing they'd be looking at.
Because
Because it's on.
it's in everything.
All living things because it's what makes
All living things.
We eat strawberries. There's DNA in strawberries.
We
Boom.
eat strawberries.
So when scientists are able to remove DNA, they can find out species of what else? So why do you think it's important for scientists to be able to extract DNA from organisms?
Scientists would be able to like clone an organ if, like in case of an accident, like right now 3D printing, something like that, clone an organ like in case of an accident.
Yeah.
So kidneys, everyone needs a kidney.
Yeah.
Strawberries can produce, but they have more than us because they're octoploid.
If you're looking at a genome,
Yeah.
If you're looking at just from start of gene 1 to last gene in our makeup,
Yeah.
yes, our chain is longer,
Yeah.
but there's more per cell in a strawberry cell.
Yeah, like think about it. Okay, with the strawberry,
The chains
less
themselves are smaller. Yes, there's less genes on the chains than are on ours.
right, because they're more complex.
Yes.
Living things.
Yes.
I think
There's more actually jammed in there,
Yeah.
but it's just a lot more chains. Does that make sense?
Yeah, because that was just in a clump. Imagine if we just took the DNA, we just found it, like, stretched it out. I know, that's what I was thinking.
But
like, super long.
but keep in mind, there's bunches of strands in there. It's not just one
One
strand,
strand,
right?
yeah.
You see what I'm saying?
Uh-huh.
So if you were to pull out an individual strand and hold it up and then compare it to an individual human strand, the human strand is going to be longer, but there's just more strands in the strawberry.
Maybe that's what I was talking about. That's cool. I was talking about how big our is. You're kind of smart, Sarah. Yeah, I kind of know something every now and then.
All right, good job, guys.
What's that called? It's kind of hard to read.
So real quickly,
For number 3, explain what happened in the final step when you added the ethanol to the strawberry extract. You know?
Yeah. When we put the ethanol in, like it all like came up, like it looked like a bunch of bubbles came up on top of the strawberry. And when we stuck the, I forget what it's called, the red loop thing in, that like all clumped together immediately. Like.
all right, for your exit ticket before you leave today, either on a sheet of paper, or if you want to go back to the bell ringer again, I just want another answer to that question that you answered first in the bell ringer. So I can see whether you got what I wanted you to get out of this or whether we need to go back and look at it again, okay? All right, so either on a sheet of paper or just go back and do another form on the bell ringer for me. Either way, just make sure on the form you put, by your name, put the number 2 so I know it's the exit ticket as opposed to the bell ringer. We good? All right, thanks guys.
Put 2 next to your name?
Yes.
Oh, no, that's my paper. No. Okay, so.